In this laboratory exercise, you're going to be investigating two things: osmosis and dialysis. Osmosis occurs when water moves through a membrane, and that's the only thing that can move. So we're talking about, in our example, we're going to use living cells. So that's just water movement. Dialysis is a process of selectively passing molecules through a membrane. So if you have any relatives who have to undergo dialysis because their kidneys are failing, then you know what this is like. And it's not a pleasant experience because you have to sit there for several hours as your blood is pumped through a machine, and it is pumped through a membrane that is submerged in a solution that is specifically designed to discourage the movement of some molecules and encourage the movement of waste molecules, like urea, for instance. So dialysis is selective. And we're going to have two processes here. One, osmosis with the potato cores, and the dialysis actually uses dialysis tubing. So the difference is this is semi-permeable membrane, and this is selectively permeable membrane. Let's see, what else can I say without taking too much time? It's going to, so you're going to start on this one, and your potatoes have to sit in the solution for 20 minutes. So while they're doing that, you go to this one. That's Roman numeral 1. This is Roman numeral 2. And then you get this one started, and you go back to this one after 20 minutes. So somebody's got to start your watch, or a stopwatch, or some type of timer, so you'll know when to take your cores out of the solution. And what kind of data are you collecting about the cores? Well, there's a data table. You're getting qualitative and qualitative information. You want to know the mass of the potato cylinders before and after they're treated. So in order to do that, you've got to sort of tap them dry with a paper towel, right? You don't want to take the core and have a bunch of solution hanging with it. You want just what's been taken up in the cells of the potato. So that's one caution. The other is you need to measure length. Right? There should be a ruler in everybody's bucket. Measure length before and length after. And I'm looking at your data sheet. And then, what are your qualitative observations? Right? Is it firm? Is it pliable? Use your own words. Okay, that's for part one. Part two, you're going to do the Benedict's test and the iodine test on solutions to see what the response is. Right? This is you're gauging. This is like calibrating your eyeballs. What are you looking for? And that's the first one. You're just going to do the pure solutions, and then you're going to take samples of the solution that surrounds the dialysis tubing and see what came through. Okay, so when you pull the dialysis tubing out of the big tube, the test tube, which are over on that cart, you want to be sure that none of the inside components of the tube spill out, right? If you're going to take a sample inside the tube, you should move it over into another beaker and then open it up. Otherwise, you might spill some in your tube and ruin your test. Okay, just those are words of caution. I'm going to put a, see, we've got 3 groups. I've probably got room if I'm careful. Put all your data up here so I can get a snapshot of all the results. Okay, so Prelab. Tell me how to make a 0.9% sodium chloride solution. This is the first question. 0.9% sodium chloride solution. Let's see, that's 1. 0.9% sodium chloride. What have I always said about percent? It's a dimensionless number. So we have to know what that percent means. In this case, it means mass volume. So a 0.9% solution is 0.9 grams per 100 milliliters. That's the 100 parts. This is the 0.9 parts per 100 parts. So it doesn't have to be exact. That's why instead of 100 milliliters of solution, we're going to use 100 milliliters of water. Right? So it won't be exact, but it'll be close enough. Hopefully. So what does the method say you need to make? Well, that question says 50 milliliters. So if you're going to make 50 milliliters, it's a ratio, isn't it? So how many grams go in 50 milliliters? 0.45 grams per 50. Okay. Part 2. We got to make, that's an isotonic solution. Oh, we skipped hypotonic, right? What's our hypotonic solution? Distilled water. You don't have to do anything with that. Distilled water. So you're going to have a potato core go in distilled water, one go in the isotonic solution, and one go in the hypertonic solution. Our hypertonic solution is going to be 20%. Right? In 50 milliliters, so that would be 10 grams per 50 milliliters. For the same reason as this one. Okay? We don't have to do the other. Explain what is an isotonic solution, hypertonic, and hypertonic. So let's see. That would be 2. 3. Describe the function of dialysis tubing. I mentioned earlier, dialysis tubing is differentially permeable membrane, or selectively permeable. That's another name. Selectively, either one works. Selectively permeable membrane. And its purpose is to restrict the movement of basically size molecules, but it can be charge related. This dialysis tubing that we're going to use is cellulose based, and it's designed to permit the passage of anything smaller than about 12,000 Daltons. Daltons is a term for the size of macromolecules, so don't worry about what it means. But anything smaller than that will pass through, and anything larger should be retained. Okay. So the purpose of dialysis tubing for us is just to restrict the movement of large molecules. But dialysis tubing for your patient does what? It allows waste materials to pass through the tubing and the good stuff to stay in your blood. Okay. And they do that by selecting what is that solution made of around the tubing. Right? If the tubing is made of, has in it various components like simple sugar, like glucose, then it'll be at a certain level, an ideal level. And then as your blood passes through, there's a concentration gradient that's set up. And if your blood has too much glucose, some of it will move out. But if it has too little, some of it will move in. Right? It's a balancing act. Okay, that was 3. How about 4? Benedict's. What's the purpose of Benedict's solution? Right. Well, if you haven't read about it, you don't know. Benedict's solution is used to detect the presence of glucose in our case, or actually it's based upon what's called reducing sugar. A reducing sugar is something that will reduce a compound. And we haven't covered that yet, but in chemical terms, reduction means it transfers electrons to something. It reduces it. As opposed to oxidize it, where it takes electrons from it. So that's Benedict's solution. Iodine. 5. What's the purpose of the iodine solution? To detect starch. Okay. Now, the chemical reaction that goes on that gives you an indication of starch is fairly complex. But all we need to know is, what does it do? And it detects starch. And it's very, very sensitive. Just tiny amounts of starch will give a very noticeable color change. If there's a lot of it, it's going to be almost black. If there's just a little bit of it, it'll be more light blue. And you're going to see the difference when you run your first tests with the pure substances and get an idea of what you're looking for.